So the second part is about antibodies, antibody techniques. So we'll talk about ELISA, EIA. We'll talk about Western blot. So all these are using the specific interactions of antibodies against an antigen or a protein or any molecule with a particular shape, right? So this is a diagram showing you the immune response where you have the B cell here, and the B cell is responsible for generating antibodies against a pathogen or any foreign material that gets into your circulation. Right? So you have a virus like COVID-19, or you have a pathogen like bacteria that gets into your body. And these are considered foreign, and then your body will interact, will try to protect you from these foreign molecules by producing antibodies against it. So that eventually the B cell, there are many different B cells in your body. Each of this B cell is responsible for producing one specific conformation that can recognize one specific antigen. Okay, so you have COVID-19 gets into your circulation as an example, and that will interact with like 3, 4, or 5 different B cells. And these B cells later on will be activated to generate memory B cells by reproducing itself. So one B cell can reproduce itself into many B cells, producing the same antibodies against the pathogens. And this is the immune response that protects you. And antibodies will interact with the T cell, T helper cells to attract macrophage. And later on, sorry, it's the plasma cells that generate the antibody. The memory B cell later on is responsible for, sorry, I mixed up a little bit. The memory B cell is for the memory, so that when the pathogen be invade again into your body, then it will produce the antibodies very quickly, protect you. But anyway, the antibody response is for, it's your body trying to protect you from foreign molecules invading the system. Okay, so this is the antibody structure consisting of 4 polypeptide chains. So 2 light chains and 2 heavy chains. And they are 2 heavy chains are exactly the same, 2 light chains are exactly the same. Okay. So they're linked by 4 disulfide bonds. So you can see that this is 1, 2, 3, 4 disulfide bonds. So from this diagram, immediately you can tell me that there are 2 different ends to this triangular structure, right? So this end, the heavy chain, 2 heavy chains here, is called the FC region. And there's only 1 of them. But these 2 ends, with 1 light chain and 1 heavy chain, and there are 2 of them, right? So there are 2, this is called the FAB region, and then this is called the FC region. And remember that very clearly. FAB region is responsible for recognizing the antigen. The FC region is the same for all the antibodies produced by the same species. Okay, so FC region is identical for all the antibodies, but the FAB region is different. And that's why each of the B cell can generate a particular antibody that can recognize a specific antigen. Okay, so FAB region is a variable region, and FC is the same for all the antibodies produced in the same species. Okay, so this is a diagram showing you. Okay, so you have different B cells, and these B cells will have the antibody presenting the cell surface, and they're all different. The FAB region, they're all different. And then this is the antigen, and this is the one. This is the one that is able to interact with this antigen, and later on producing the plasma cells and the memory B cells, generating many antibodies against this antigen. So this is a diagram showing you there are many different B cells, but only this one in this particular diagram can interact with this antigen, giving rise to a lot of antibodies against it. Okay, so now you can imagine that if we have a protein and you use centrifugation technique, chromatographic technique, you purify your protein to homogeneity, and then you inject this protein into a rabbit or into a mouse or into a rat, doesn't matter, or donkey, horse. This protein is considered foreign to that animal. And then it will generate antibodies against that protein. You can take the serum out from the animal and can purify the antibodies, or you can use the serum directly for detection of that particular protein. Okay, so this is the basis for all antibody techniques. First of all, you purify a protein, you inject into rabbit as an example, and then the rabbit will generate anti-serum against that protein. You take this, you bleed the rabbit, take the serum, and use it for detection of your protein of interest in many different techniques. Okay, and this one is called immunohistochemical staining or immunocytohistochemical staining. Okay, basically, what you have is you have a section of cells. So you take, so imagine that you're working on a hormone, like my lab works on hormone. And this hormone is produced in the pituitary. Okay, pituitary is a small little gland called the master gland on the top of your nasal cavity. And you take the gland now, you section it really thin. We're talking 5 to 10 microns. And then you can use the antibodies that you generate to detect a presence of a protein in these sections. Okay, so you have the section, you have the primary antibody that you generate. And then afterwards, remember there's an FC region. FC region is constant for, it's the same for all the antibodies being generated. So for the sake of simplicity, some lab that can sell a secondary antibody that can recognize this FC region and also with an enzyme attached to it, you buy from that lab and then you put some enzyme substrate for detection purpose. And then you can detect the primary antibody, which interacts specifically with the protein of interest. Right? So this whole procedure requires sectioning, and then you block with non-specific protein, incubate with the primary antibody, and then in this case, a secondary antibody that recognizes the FC region of the primary antibody, and is conjugated with an enzyme called alkaline phosphatase. And then you can use a chromogenic, meaning color generating substrate, BCIP-MBT, so that some color will be developed and they can detect your protein of interest. Okay, so this is the kind of result that we're looking at. So this is a section, and it's counterstained with hemotoxin. So all the cell is stained blue, but in this case, the Ki-67 primary antibody is used for the detection of the Ki67. And then you have the alkyne conjugate secondary antibody with NBT-BCIP for the detect. So as you see this purplish brown color being developed, and these are the cells expressed of your protein of interest. Okay, so it is called immunohistochemical staining. So if you look at individual cells, there's immunohistochemical staining. Okay, so now because we can attach a conjugate enzyme to the secondary antibody, you can also attach a 404. Okay, a protein or a chemical structure that can be activated by laser. And when it relaxes, it can give out a color you can detect. Okay, and this is immunofluorescent staining. Okay, basically the idea is that if you have a 404, that you use a high energy light, in this case a blue light, you excite the 404 to this excited state, and from that it will be relaxed to the ground state of the excited state, and then from there it will emit light, in this case a lower energy light, a green light, back to the ground state here. And you can see that this technique is a lot more precise in terms of locating the protein of interest in the cell, because you can actually see, in this case, you can actually see the fibers. So the green color here is the cytoskeleton being labeled with the antibody. Okay, so again, primary antibody that interacts with this cytoskeleton, secondary antibody conjugate with 404, and then you can use laser to activate 404. You can see the precise location. Very, very nice structure being observed in this kind of staining techniques. So very powerful technique. And you can also label the DNA, in this case, with a blue color, the blue 404, and then you can use two different lasers to activate the two 404 and see them all together. And another example here, again, this green color is labeled with anti-alpha tubulin, and then you can also label the chromosomes with anti-CDC6 antibodies. Okay, so again, a very nice diagram showing you how you can use this immunofluorescent labeling technique to see, to observe what is happening in the cell, in this case during mitosis, during cell division. You can see the sister chromatid lie in the equator region, ready to be separated by the simple spindle fibers. Okay, so we have talked about using antibody for detection of protein on sections, like 5 to 10 micron sections, to see the location of the protein in a tissue, right, in cells of a tissue. We can also use immunofluorescent labeling to see expression of certain protein within a cell in different cellular structures. Right? So very powerful technique. But majority of my lecture will talk about a more simple technique called ELISA. So in your lab, I'll give you some antigen that you later on will perform ELISA. It's a very simple technique, but it is used commercially extensively. I'll give you some example. We'll talk about the pregnancy test. We will talk about that you can actually buy from the pharmacist. We'll talk about the COVID-19 neutralizing antibody test. We'll also talk about the mad cow disease using ELISA as a technique for detection. So these are some of the examples that are being used, and all because of the fact that we can generate antibody against the protein. Okay. So before we talk about ELISA, which is short for enzyme linked immunosorbent assay, so enzyme linked immunosorbent assay, we have to talk about the different forms of different techniques of ELISA. One is called the direct assay, the other one indirect assay, and then you have a capture assay or sandwich ELISA. So it's three different types, and they're easy to understand. Don't worry. Direct assay is when you have antigen coated on a plate, and then you have the primary antibody already conjugate with an enzyme. So you can conjugate the primary antibody directly with an enzyme, but it's a lot of work to do it. And commercially, of course, companies can do it, but in the laboratory, I cannot conjugate all my primary antibody with enzymes. So I use secondary antibody to detect the primary antibody. But commercially, because they're producing a lot of the ELISA kits, like pregnancy test kits, then they can directly label the primary antibody for detection purpose. Okay, so antigen primary antibody already conjugate with an enzyme. You can therefore directly test the presence of the antigen and the primary antibody complex by putting in substrate and see a color. The second one is more popular. It's like in my lab, every day we perform this assay. It's indirect assay where you have an antigen attached to a plate, in this case, ELISA plate. The primary antibody detected by secondary antibody conjugate with an enzyme and then substrate turn into color. Okay, so this is a more common technique in the laboratory, but this is more for commercial purpose. The third one is called sandwich ELISA, and it could be a direct or indirect sandwich. So if you have a capturing antibody, you conjugate, you coat the plate, the ELISA plate with, and this will be responsible for capturing the antigens. And then you have another primary antibody that can recognize the antigen again. Then you use a secondary antibody for detection. But you can also directly label this primary antibody for detection, conjugate with an enzyme for detection. Okay, so it could be either direct or indirect sandwich. Okay, so there are only three different ways for detections. And oftentimes, you will see a color change later on, either in solution or in Western blot or in cytochemical staining. You will see actually the color deposit onto the cell or the material for you to detect. Okay, so a very similar technique. This is the ELISA technique. This is the ELISA that you will do in your laboratory. So in the student lab, we will use the indirect assay for the detection of antigen in the sample. So again, I'll give you some antigen, you mix it with your friends a couple of times. And then this is the ELISA plate. And then you will coat the antigens onto the plate. And then you use the primary antibody for detection of the antigens. And then you use the secondary antibody for detection of the primary antibody, FC region of the primary antibody. And finally, you use an enzyme substrate to develop a color. Okay, so you can see a blue color in the end. Okay. So this is the enzyme substrate is TMB, and the enzyme in this case is horseradish peroxidase. Okay, so it will develop blue color. You can see the blue color in the end of the day. So the immunoassay can be used for tracking outbreak of disease, like HIV, SARS, COVID-19, smallpox, anthrax. All these outbreaks, you can actually use the ELISA assay to track them, okay? And basically, ELISA can either be used for the detection of antigen or detection of the antibodies in serum. Okay, so imagine that someone is infected with COVID-19. Then this person will have the antibodies in the serum to COVID-19. You can use ELISA, the sandwich ELISA. You can use ELISA to coat the plate with the antigen. You can capture the primary antibody in the serum of the patients to detect the antibody. Okay, and then you can also detect the antigen by sandwich ELISA, where you coat the plate with an antibody to capture the antigen, and then you can evaluate whether that person has the antigen or not. But nowadays, of course, in addition to antibody techniques, we use mostly the RNA test, right? So you use reverse transcription of the RNA back into DNA and use PCR. And you use a quantitative PCR to also measure the virus, whether you have a lot of virus developed in the system or not. Right? So both antibodies and also RNA. You're supposed to turn off the phone during lectures. Anyway, so the ELISA technique and also the RNA test is used nowadays for the detection of the COVID-19. And in the past, SARS, you can use it for, I'll go through the pregnancy test with you as well. Okay, so this is the pregnancy test. And anyone have used that? No, right? I'm sure you have buy this test for your friends, right? So you're your friend, you need that test. So this is a test of, you can buy this in any pharmacist nowadays, and then use urine. And because pregnant women during even early pregnancy produce a hormone called HCG, human chorionic gonadotropin, HCG. So this is HCG, okay? So I don't know why. Anyway, so what you do is you dip the sample with urine, and then this is the positive test result is when you see 2 red lines, and then this is a negative test result is when you see only 1 line, which is the control line. Then T is the test line, and then you have the control line. So the idea is that there's a color antibody that will migrate together with the analyte that is present in the urine sample. And then this labeled antibody, is the red color, will travel along. And then when there's an HCG in the urine, this primary antibody is able to interact with HCG. And in the test line here, you will have another antibody that can recognize the HCG, and this is a direct sandwich ELISA. So you will see the red color, the labeled primary antibody forming this line here. So you also need to know whether the test is good or not, right? So you need a control. So you make sure that there's some color antibody that's traveling down this test here. So in this, you have also a control line where it is coated with an anti-mouse IgG antibody that can recognize the Fc region of this color antibody, so that you will need to see a red line here in the control line to make sure that if you see a red line in the test line, then this is really positive. Okay, so if you do not, for example, in a negative test result, if you do not see a red line in the C line, that means that there is no color antibody that is traveling down together with the urine sample. Then this test is not valid. So again, the test line consists of a sandwich where you have an antibody that can recognize the HCG, and HCG already interact with the color antibody to form a sandwich, a direct sandwich. And the control line, you have another antibody that recognizes the Fc region of the color antibody, so that you make sure that there is a color antibody that travels together with the urine sample down this test to form a red line at the control line. So this is the pregnancy test. Okay. So if your friend asks you now, you can explain to them how it works. Okay, so this is the rapid testing for the presence of COVID-19 neutralizing antibody in serum. And I basically just download this diagram from internet. And this is produced by a company called Zero Fresh. Okay, so the idea is that when someone is infected with COVID-19, or when someone's immunized with the COVID-19 vaccine, then this person will produce antibodies. Some of the antibodies are very important for the protection of the patient, right? So of the person, particularly for vaccine. And this is called the neutralizing antibody. So the neutralizing antibody is capable of interacting with the ACE2 receptor on the human cells. And because of that, therefore, it is able to interact with COVID-19 virus and block the interaction of the spike protein with the ACU receptor. So it neutralizes the COVID-19 virus and protect the person from infection. And that's why we need to inject, we need to vaccinate ourselves with the COVID-19 vaccine, right? So this is the reason. So to detect whether you have the presence of the neutralizing antibody, basically what you have here is that you have the ELISA plate coated with the spike protein. And then if you have the neutralizing antibody, then it will protect it from binding with the ACE receptor. And because of that, then there will be no color being developed. But without the neutralizing antibody, then the ACE is capable of interacting with the spike protein. And then if you use a secondary antibody for detection, then the detection complex, you can therefore see you put in the homogenic substrate, then you can generate color. Okay, so this is the principle for detecting whether someone produced the neutralizing antibody or not. So you need this as a control, and you make sure that in this well, there is no color being developed to see that there is actually neutralizing antibody produced in the system. Okay, so one more example on ELISA, mad cow disease. Okay, so mad cow disease, there are ELISA tests developed for this BSE, bovine spongiform encephalopathy. Okay, so it is because of the presence of a prion protein that is being mutated. So this is a normal prion protein with a molecular structure like this. You can see the alpha helical structure this way. But because of a point mutation of this protein, then there's a complete change in conformation of the protein. And this protein, therefore, is responsible for the mad cow disease. Okay, so for that reason, therefore, you must find a way to detect whether you have the normal prion protein or the disease prion protein with a structure like this. And the easiest way, of course, is by ELISA. And it is because this protein here, the alpha form, the normal prion protein, is proteinase K sensitive. So you can use proteinase K to digest this protein. But because of the conformational change of the protein with the mutation, then this protein is proteinase K resistant. You can therefore use this property to distinguish the two different forms of the prion protein by using brain extracts from different cows. Right? So, this is the disease versus normal prion to prepare the sample. And then afterwards, you use proteinase K, which only digests the normal, but not the disease prion protein. And then you use ELISA tests for the presence of the prion protein afterwards. And since 2004, there were more than 300,000 cows being tested for in the USA alone for this mad cow disease. Okay, so this is a diagram showing you exactly what is being done in the laboratory. So you prepare the brain tissues from cows, and then you homogenize the tissues, digest with proteinase K. Remember that normal prions are being digested, and disease prions are resistant. And then you denature proteinase K, and then perform ELISA test. And this is the kind of result that you will see. This is a typical ELISA plate consisting of 8 times 12, 96 samples per plate. Okay, so this is a standard format used in all the laboratory across the world. And therefore, you can imagine that we can devise machines that can do all the procedures for us. Okay, so put in sample, put in different test solutions, all automatic nowadays in big companies. So they can handle a lot of samples. So usually we do in triplicates to make sure that there are no false positive or negative. So this is a positive control, 4, 5, 6 negative control. And then these are the test results. You can see that if you see a blue color developed in all the 3 wells, meaning that the brain sample from that particular animal will contain the disease prion protein. Okay, so this is a typical test being used nowadays for the screening of the mad cow disease.